How are you?
Good.
So today our speaker is our very own Anastasios Matzavinos,
Thank you.
who was supposed to be speaking in person, but Chilean government has managed to postpone his arrival just a little bit further, a couple of weeks. So it is a pleasure to introduce Anastasios, who will be speaking today about data assimilation, uncertainty, quantification, and molecular dynamics. Anastasios was a faculty member at Iowa State and at Brown before joining us at the EMSA. And you obtained your PhD, I don't know where.
That's in Scotland. That's the University of Dundee in Scotland. So yeah.
Oh, okay. There you go. So, well, without any more delays, I introduce you. There's no applause, but just go ahead and start and share your screen. Give me 1 second. I'll give you.
I've given permissions to share my screen, and that's all I need.
There we go. Now you can share your screen.
Give me just 1 minute to set this up. Share. Let's do that. Okay, and I think we are good. Let me move this window around. All right, so let me start by saying quiero hablar español, pero mi español no es muy bueno. So I think I got that right. So I will stick with English today, but hopefully sometime in the very near future, I will be able to give a talk in Spanish. All right. So the talk is about molecular dynamics and data and how you can bring them together. All right. How you can do data assimilation, you can integrate data that you may have, or you can design molecular structures on the basis of existing data by utilizing molecular dynamic simulation. So let me start by saying, oops. All right. Okay. So let me start by saying that this is work that I did with a couple of former students of mine, Clark Bowman and Karen Larson. They both have been very successful. Clark got, after he graduated with me, he got a postdoc position at the University of Michigan, and then he moved on to a tenure track position. Karen was even more successful. Karen defended her thesis, I think it was April 2020, just a week before Brown University closed down for COVID. So just at the nick of time. All right. So Karen got multiple offers from academia. She wanted to, for her own personal reasons, she wanted to move to a position in the industry. So she turned down the positions in academia and she got a position as a researcher, as a research data scientist at Disney Plus, the streaming services. So she has a pretty good salary. And the reason why I'm mentioning this is because I actively look for new students. All right. So if you are a graduate student and you have any interest in molecular dynamics, how you can integrate molecular dynamics with data science and so forth, talk to me. All right. We do. I have a new grant. There is funding. So there are many projects that might be of interest to you. All right, so let me start by giving you an idea of the kind of molecular dynamics that my group back at Brown was doing. I hope this actually worked. Can you see the simulations? Does this thing work smoothly, or you can? All right, perfect. All right, typically a molecular dynamic simulation consists of some sort of a particle representation of a molecular structure. So what you see here on the left, I think, yeah, on the left is a lipid bilayer membrane, right, which is propelled forward by some polymerizing filaments. So if you're a molecular biologist, you can think of the actin cytoskeleton polymerizing and pushing forward a lipid bilayer membrane. And so every particle in this simulation corresponds to a stochastic differential equation. You need stochastic differential equations because thermal motion is important in this kind of nanoscale simulations. Let me stop that because it's kind of, all right, so it's kind of distracting. Now, the thing that you don't see in the simulation is the solvent. You have no water molecules around. And so this patch of a lipid bilayer over here is something like 50 nanometers by 50 nanometers. And if you wanted to have a full-blown molecular dynamic simulation that tracks every possible water molecule in that little cube over here, that would be infeasible with current technology. All right, it simply cannot be done. Right, so yet we have to do it. All right, and we do it. All right, so we can simulate molecular dynamics. We can simulate molecular structures with the solvent present. So how do we do it? We don't track any possible water molecule. We cross-grain the molecular dynamic simulations. So the basic idea is that instead of following or tracking, if you wish, every possible water molecule, you track collections, lumps of water molecules. All right. So just to back up a bit, so basically what a molecular dynamic simulation is, and that's a cartoon over here on the left, right? These blue spheres are supposed to be water molecules, all right? Basically, a classical molecular dynamic simulation is just Newton's law, right? F is equal to ma for every particle. And you have what you have here, for example, in this cartoon is a polymeric structure, a polymer and water molecules. And these interact according to certain potentials that come from chemistry. Usually you employed something called the Lennard-Jones potential or family of potentials. All right, so you see that the forces here are conservative, they come from the potential. All right, again, that's super duper expensive. So that's something that you can do only for small structures. On the other extreme of the scale, you can talk about Brownian dynamics. Now, that's a very restrictive approach because here you assume that the solvent is there, but it does nothing. You don't have any kind of flow. All right. And the idea here is that, all right, you prescribe the collisions of the water molecules with whatever structure you're focusing on indirectly through basically some random variable, this eta over here. Eta sub I corresponds to every particle I over here. Right, in that polymeric structure, and that eta is white noise, and it's supposed to represent the collisions of that structure with the water molecule. So you see, you have some sort of a viscosity term here, which is opposes, it's proportional to negative the velocity of every particle. That's a viscosity force, and you have some forces which are random. Now, I write this as a differential equation, as an ordinary system of ordinary differential equations, but mathematically speaking, that has to be set up properly. Right, so white noise is not a function; it's a distributional squads. All right, so you can perceive white noise as a weak derivative of Brownian motion. All right, in the sense of distributions. So that's something that we did in my class last semester. How you set up systems like that properly in a weak fashion. In the sense of distributions, you need the notion of a to integral and so forth. The problem with that is that you cannot do mechanics. All right, you cannot have any notion of flow here. Right, you don't have any exchange of momentum. This is supposed to be some simplistic approach where basically the solvent is stationary. All right, so an approach that, yeah, go ahead.
Can I interrupt 1 second? Can you go back to the last slide?
This one? Yeah.
So the reason it's expensive is because that potential is basically relating every single particle with every other particle, right? Is that the reason?
So you can go about that by basically having sub cutoffs for the potentials, right? So you say, all right, so if you have a potential, if you have two molecules that are far away, you have a very weak force. So basically you can have a neighborhood where this force is present, right, with neighbors, and then you can have some cutoff outside that neighborhood, and that contributes to the complexity. But the mere complexity comes from the fact that you have a lot of molecules. All right, and even in small neighborhoods, if you track every water molecule, even in small neighborhoods, right, you're going to have a lot of interactions. All right, so that's where the complexity comes from. You don't have to have interactions for every possible molecule because you can say, all right, outside the radius, the force is 0. I impose that, right? But still, due to the mere amount of quantity of molecules that you have around, all right, if you want to focus on a, 100 of nanometer scale, you have gazillions of water molecules. That's not a technical term, but anyway. Okay. All right, so that's the idea. That's where the complexity comes from. All right. So now, somewhere in between, you can start thinking about coarse graining, and now you say that's a very natural idea. You say, hey, all right. So here's what I'm going to do. I'm not going to track every possible molecule. I'm going to track lumps of molecules, collections of molecules. So now I'm going to have a particle simulation, particle-based simulation, where every particle corresponds to a collection of molecules. That's the idea. Now, that's an elegant idea, All right. So, but the technical aspect, the mathematical aspect of it is how do you prescribe forces to these collections of molecules? So as when you have a lot of them, when you upscale the system, at some asymptotic limit, you obtain the correct constitutive relation. If you want to obtain Navier-Stokes, you would like your stress tensor to have pressure and viscosity, right? So again, it's an elegant idea, but you have to have a methodology for prescribing appropriate forces that they would capture the appropriate constitutive relations. And there is a lot of analysis that goes into that. I'm not going to go through the analysis. That's not the subject of the talk, and the analysis is not mine anyway, but it exists. All right. All I'm going to do right now, I'm going to give you a 5-minute introduction to this coarse graining approach that is called the dissipative particle dynamics method. I'm just going to describe the forces. All right, and ask me later on how these forces were figured out. All right, in terms of giving you the correct constitutive relations. All right, so you have 3 types of forces. All right, the forces are center to center, and there are forces applied to your particles, which again are collections of molecules. All right, and you have conservative forces, dissipative forces, and random forces. All right, 3 types of forces. All right, so the conservative forces are different than the type of forces that come that are obtained in classical molecular dynamics. The Lennard-Jones potentials, the Lennard-Jones family of potentials in classical molecular dynamics has a singularity at 0. Right? It diverges to infinity. And the reason for that is you don't want 2 molecules to coincide in space. Here, since your particles are collections of molecules, they can pass through each other. All right. And so the idea is that you prescribe a soft potential, which also helps computationally. You don't have any singularity. All right, so it's a conservative force. The potential, the force comes from a potential. Usually you adopt some sort of a linear force, all right? And sometimes you have a cutoff radius. So, that comes from a potential almost everywhere, except here, all right, at the corner. All right, but that's a conservative force, the purpose of which is kind to discourage the molecules, the particles to go through each other, but they can do so, all right? Then you have a dissipative force, which plays the role of viscosity. All right, so again, all the forces are center to center. So you look at the relative velocity between 2 particles, Vij, you project it to the vector that connects that particles, hence the inner product over here, and you oppose that relative velocity. All right, so that's a viscous term. All right, so, and you have a weighting function in all of these forces, you have a weight function, which you can adjust so that you can get the kind of constitutive relation you want at some asymptotic limit. All right. And keep in mind, look at that coefficient gamma over here. It's going to come up later on. All right. So this is this coefficient of proportionality. And then if these are the only forces that you had, conservative and viscous forces, then the flow, whatever flow you had, would die out. All right. So you have to have some agitation, all right, in the system, and that comes through some thermal forces. All right. Now these are center to center. They're not like the thermal forces in Brownian dynamics or in the Langevin equation. So you have, again, a family of random variables, 2-parameter family of random variables, psi ij. These are distributed according to white noise. All right. So again, in order to set up these systems, this white noise is a distribution, it's a bona fide distribution. All right. Alaskfart is not a function. Again, a weak derivative of Brownian motion. So you have to be careful how you set this up. All right. But the important ingredient here is that, again, these are center to center, and you impose that psi ij is equal to minus psi ji. So, with every force, you have an equal and opposite counter force. So you can do mechanics. You have conservation of momentum by imposing this for the thermal forces. It's not like Brownian dynamics. All right. So that's the setup. Now, how one would, this seems very ad hoc, right? But there is analysis behind it, and there are reasons. I mean, the forces look reasonable, but one has to convince themselves that at some asymptotic limit, you get some proper PD. All right. And you have to take my word for that. I'm not going to go through it. All right. So there are a couple of extra conditions here that you have to impose. If that's all you had, all right, you have a conservative family of forces. You have dissipative forces that subtract energy. You have random forces that on the bulk they add energy. They may subtract energy randomly, but on the bulk they add energy. So these have to be balanced somehow, and they balanced by a fluctuation dissipation relation that comes from statistical mechanics. So sigma and gamma are the coefficients for dissipation and fluctuations. All right. So sigma is the coefficient for the random forces, and gamma is the coefficient for dissipation. So this introduces a notion of temperature to the system when you do the numerics, right? So you impose that sigma square is equal to twice gamma times Boltzmann's constant times the temperature. Right, and that also imposes certain conditions on the weight functions. So that's the setup of dissipative particle dynamics, and I'm going very quickly through that because that's not my focus. Right, but that's the kind of molecular dynamics that we use. We do a lot of this stuff. We also are interested in analysis for this kind of systems. It's a huge system of stochastic differential equations. That's what you have to keep out of this. All right. I'm not going to talk about the numerics. The numerics are interested here in the sense that a lot of people use symplectic integrators for this kind of stuff. The idea is that you are not really interested in high precision per particle. You're not interested in the particles at all. You're interested in the behavior of the ensemble. You would like the ensemble to have some nice properties, like, for example, for symplectic integrators, you would like to preserve the two form that is given by momentum and position. All right. So
sorry, I'm sorry, that's what I had to ask you about that.
Ask me, but go ahead.
But then I didn't see. Okay, so my question
that's
is,
not a symplectic integrator, if that's what you are going to ask. But
no, I mean that's right. But then you talk about symplectic, but it's not clear to me that the system is going to be Hamiltonian then.
Right. So we're going to come back to that. All right. So that also has to do with the weights that you're using, right, in order to achieve the appropriate. So I'm not saying that symplectic integrators are appropriate for all types of dissipative particle dynamics simulations.
Okay.
Right. But for some weighting functions, you have the structure that would allow you to, that will make the symplectic integrator a good choice. All right. So ask me towards the end because I want to go through some material. All right. So the way that we use this stuff, so just to give you a good indication for how these things are used, this is a project that I had with Derek Stein. Derek is a guy who does nanofluidics at Brown. So Derek, this is a project that I talked a little bit about in my job talk, so you may have seen this. So the idea here is that Derek manipulates DNA macromolecules. Right. So the idea is that when you have a DNA macromolecule embedded in a solvent, usually in its native state is all balled up. And the idea is how can you unfurl it, how you can stretch it. All right. And so there are many ways to do that. All right. Usually the methods of preference are through nanofluidics. So the idea is that you squeeze the DNA molecule between two glass plates. All right. You impose a pressure difference. You have a pump that imposes. So these are the two glass plates over here. They're at a distance of something like 100 nanometer, glass plate from glass plate. You squeeze the molecule and you add some nanowells at the bottom plate. And then you impose a flow, right, with a pressure difference, with a pump. And the idea is that as the DNA molecule moves along this channel, it will expand dropically above one of these wells. It will get trapped and the flow would kind of unwind it. All right, so that's one way of unwinding, a kind of stretching DNA molecules. Now, Derek wanted to measure forces in this system. All right, this is a very confined system. You cannot put optical tweezers in there. And the problem is that if you do that more often than not, this is what you see here on the right, the DNA molecules just jump from well to well. They don't really unfurl. You really have to have very specific parameters here in terms of the depth of the wells, the distance of the wells, so that the DNA molecule unfurls. And that's what we did. We designed some of these devices with the Bayesian methodologies that I will be describing to you. All right, so so he came to me and said, can we measure forces? All right, and how much time do I have? And so I said, and the other thing that he observed was a very interesting behavior, which was very interesting for him. Let me skip that, actually. And I will not tell you about the behavior that he observed, but we're able to capture with our simulations. Anyway, I'm giving a very short introduction to molecular dynamics because I want to talk about data today. All right, so here are a couple of simulations that basically try to capture what he was saying. Let me tell you about this behavior, what he saw. So the idea is here, if you basically look at all these molecules that jump from well to well, and you measure the average velocity, so these move upward over here, right? And you see these kinds of slow, and you have a fast one that moves faster, it gets trapped, and so forth. All right. The average velocity, if you do some computation for the average velocity, would give you an average position. And then you can start talking about the deviations from that average position. All right, so basically you say here's the average velocity times delta t, and for every molecule I'm looking at the deviation of that molecule from the average position. So that scales according to delta t, the time elapsed, right, between measurements. This exponent over here is equal to 1. All right, so that's a parabolic scaling for the diffusion coefficient essentially. And basically what you observe is that the coefficient of proportionality kind of depends non-linearly with respect to the velocity that you impose on the flow. And that's something that we wanted to understand. All right, so basically here you have two devices. One has nanowells, the blue curve is for nanowells of 100 nanometer depth, and the red is for nanowells of 200 nanometer depth. So, you know, deeper wells means that you capture the molecule, the DNA molecule stronger because it expands more. Remember that molecule is kind of squeezed between the plates. All right. And so we would like to kind of understand that kind of non-monotonic behavior. So basically we started setting up the system computationally with dissipative particle dynamics simulation. So what you see here is a fully three-dimensional simulation viewed from the top. All right, so these gray squares represent the wells. All right, so here I have periodic boundary conditions on the left and right, all right, and I have a reflective boundary conditions on the glass plates. And the idea is that I visualize just a column of water. There's water everywhere, but I just visualize a column of water. Everything here is set up, the proper viscosity at the nanoscale level, etc. All right, so you see a little bit of circulation, all right, above the nanowell, and you see this kind of wedge forming for the water. So if you put some sort of a polymeric structure like a DNA molecule in this kind of configuration, this is for parameters, this simulation is for parameters where the DNA molecule will start extending. All right, so you see that the molecule kind of gets trapped with the flow. It does this kind of follows this wedge and kind of starts getting unfurled. All right, so now what we wanted to do in order to start basically studying this kind of non-monotonic behavior that he has observed for the diffusion coefficient, for that dispersion coefficient, is that this is a very simple system if you think about it. You have just water and the DNA molecule in terms of the mechanics, right? In terms of water, you know everything. You know viscosity in the nanoscale, right? You can set up your DPD methodology very, you know, precisely. Now, the only thing that we were not very familiar with was basically the mechanical properties of the DNA macromolecule, because these change with respect to the sequence of nucleotides you have, the DNA molecule. All right. And so we're able to infer this by basically looking at the fluorescence and inferring the radius of gyration of the molecule. So you see, if you have a low bending stiffness for this molecule, it will have a lower radius of gyration. It will be more compact, the molecule. If you have a high bending stiffness, all right, it will not bend as much, all right, and so it will be more extended. And you can infer that from the fluorescence that you see in his experiments. All right, so then the idea is, again, these are three-dimensional simulations. You just see them from the top. There is water everywhere and so forth, right? So, but I'm just visualizing molecules. So if you run a simulation for the parameter regime where the diffusion coefficient he was measuring was not, it was suboptimal, let's say, it was below the peak, all right? And you measure it for a velocity, which in his experiments, basically he achieves optimal peak dispersivity, peak diffusion coefficients. Let's see if you can spot what is the difference between these two simulations. All right, so obviously the one at the bottom is going to be faster because you have a kind of a faster velocity, higher velocity. But let's see if you can see what is the qualitative difference between these two runs. All right, that could be an indication that this is the causative factor of the higher diffusion coefficient. Let me run it. Oh no, where is the play button? Here we are. All right, so this moves around. All right, so it starts jumping from well to well. There are periodic conditions on the left and right, so the molecule is going to be reintroduced from the left. All right. Do you notice any difference between the two? I mean, the bottom one is faster, but that's to be expected because you have a higher velocity. So you see, the top one does one jump per time. The one on the bottom does a couple of jumps per time. Is that clear, right? With the periodic conditions, right? So you have the bottom molecule, the top molecule jumps, it gets trapped, and then jumps again, it gets trapped. This molecule over here does two jumps per time. So he does one jump, he extends more, and then does two jumps per time. All right, so that we found interesting. interesting. And we thought, let's do a little bit of analysis to understand this. All right. So it seems like higher diffusion coefficient. What do I mean when I say diffusion coefficient? I mean, if you send a bunch of DNA molecules through this flow, all right, and you see the basically standard deviation around the mean, that standard deviation extends, it seems to be larger when you have multiple jumps. And we thought, all right, so let's analyze this a little bit with a toy model to see if we can get a result like that. That's what the simulation seems to be indicating. So, by the way, we measured basically the diffusion coefficient in the simulation, and it matches what he was observing, right, for the 100 nanometer wells. All right. But then we wanted to understand why this is the case. So with Alex Reutenstein, let me skip all of that, we formulated some sort of a toy model in order to do a little bit of analysis. All right, and the toy model is as follows. So basically what you have over here, you have two particles that represent the end of the DNA molecule, and these perform biased random walks. All right, so they jump from the lattice points here in these random walks correspond to nanowells. All right, and you have some, these are continuous time random walks. So you have an infinitesimal generator that is provided by certain transition rates. So here there are A, beta, lambda, and mu. Obviously, the higher the velocity, the higher beta and mu is. And these two random walkers are not independent. They have a mechanical constraint, which is basically the maximum length of the polymer. All right, so you have these two random walkers, and then as soon as they reach a specific maximum distance, which is given by the length of the polymer, then with probability 1, we'll move inwards. All right, so you can formulate that as a stochastic process. You have x1 of t and x2 of t. These are continuous time random walks. All right, again, they're defined in terms of the infinitesimal generator. And the idea is that continuous time random walks, the movement is given by an exponential clock. All right, every time that exponential clock ticks, then one of them is chosen and is going to move either to the left or to the right with probabilities that are determined by the transition rates. All right, and you have the extra mechanical restrictions that these always have to be within S units, because that's the restriction that is given by the maximum length of the polymer. Is that kind of clear how I'm trying to set it up probabilistically, this toy model? All right. All right, so now the thing is that we would like to get some quantity for the asymptotic diffusion coefficient of, let's say, the right end of that polymer, right? Let's say x2 of t. All right, now x1, x2 is a Markov process, but neither of them, neither x1 or x2 is a Markov process. All right, because of that extra condition that basically forces you to move in, if you think about it. All right. And I'm going to define also this notion of the configuration of the molecule, which is basically the distance between x1 and x2. All right. So what do you want to do? Basically, what you want to do is that you want to prove a law of large numbers for x2 of t that would give you the asymptotic velocity of x2 of t, and the central limit theorem for x2 of t, which would give you the asymptotic diffusion coefficient. And we would like to obtain these as functions of the parameters of the model, the transition rates, because again, that transition rate mu that I had over here, all right, essentially very indirectly, that's a toy model, we want to do analysis, very indirectly captures the velocity of the fluid. That's the idea. All right. And how do you prove, we have something which is not even a Markov process, x2 of t is not even a Markov process. How would you prove a central limit theorem for such a process? Well, you embed an IID sequence into the process. You find a renewal structure. All right, and the idea here is that if you focus on this sequence of stopping times that I call tau k over here, which are the times at which basically the configuration of the molecule reaches a specific distance, let's say the maximum distance. So I track the times that these two ends of the molecule are at maximum distance from each other. All right, so the idea then is that you can very easily prove that if you look at the differences in time between those stopping times, if you look at the intervals tau n minus 1, these are IID, these differences are IID random variables, right? They're independent and identically distributed. And so because these are IID, then the trajectory of the right end of the molecule, the increments of the right end of the molecule over those time intervals are IID. So that's all you need to prove a central limit theorem or a law of large numbers. Right? Once you have this lemma, then with very standard probabilistic machinery, you can prove that the asymptotic velocity, which is xt over t as t goes to infinity, that's a law of large numbers. It will give you the asymptotic velocity, all right, is given by this expression over here, which is something intuitively that you would expect, right? Is the average increment over the average time elapsed. That requires a proof, but it's provided by very standard machinery given by the renewal structure. You embed an IID process into x2 so that you derive the asymptotic velocity. Similarly, you can prove that this process over here properly scaled, which is basically the process x2 properly scaled, converges to Brownian motion with a diffusion coefficient given by that expression over here. Right. That requires very standard probabilistic machinery, elaborate machinery, nonetheless very standard. But notice that this doesn't do for you. This quantity now has to be computed in terms of the parameters of the model, if we want to say anything useful to understand the behavior that I was showing you in the simulations. So what Alex Rothenstein and I did, so this theorem, again, is derived by very standard machinery in probability theory, but it doesn't tell you much. This quantity cannot really be computed easily. All right. So what Alex Rothenstein and I did, we actually reproved this theorem, the functional central limit theorem for that specific system, by using a spectral analysis of the infinitesimal generator of an associated process. So ask me about that. I can go through that. This could be another talk. I can go through that in detail. And we did so in a way that we get explicit expressions for the diffusion coefficient of the limiting Brownian motion in terms of the parameters of the model. And what this shows you, just to jump ahead, right, this shows you, these are now graphs that come from these analytic expressions I just described. This is the parameter mu, right, which is the transition rate of the right particle forward, which is a proxy of the velocity of the fluid for us in this toy model. All right. And you see what you have over here is essentially the diffusion coefficient of the limiting Brownian motion, of the asymptotic Brownian motion. All right. And you see that as you increase the length of the molecule, as you increase the maximum distance permitted between these two ends of the molecule, the peak diffusion coefficient goes up, which corroborates what we were seeing in the simulations. Right? In the simulations, the diffusion coefficient was increasing, right, when the molecule was doing multiple jumps. All right, and of course, if you have a larger molecule, you can do the molecule in the simulations I showed you could do at most 2 jumps per time, right? But if you increase the size of the molecule, then you can do 3 jumps per time, etc. And the analysis tells you that this increases the diffusion coefficient. Now, you might ask, why are you so obsessed with that diffusion coefficient? The reason is because you can use that diffusion coefficient indirectly to estimate the forces in that device. And so for us, that was a good test that the forces that we're measuring with the molecular dynamics simulation were actually correct. We had to have a check, right? So, okay, I simulated that with molecular dynamics, but how do you check that the forces that you measure, that they couldn't measure experimentally, could not measure experimentally, are actually correct? And there is a very indirect approach to that by measuring dispersivity to infer forces. So we compare the forces that we compare the forces, I think that's back here. We compare the forces that you can get from the dispersivity coefficient with the forces that will go from the molecular dynamics simulations. There was agreement. All right, I'm not going to go through that. That's the types of molecular dynamics simulations that we do. All right, that completes that kind of discussion. If you're a graduate student, we have a lot of projects now, a lot of projects with molecular dynamics that have to do with the actin cytoskeleton for eukaryotic cells, etc. So we can talk about this. All right. Now, what I want to talk is how do you assimilate data in this kind of simulations? How you can use data to infer parameters. All right. So how you can set up inverse problems with this kind of molecular dynamic simulations. All right. So roughly speaking, right, what I just described is with a kind of a simplistic error prediction model, a simplistic statistical model, is the following. Right. So I have a model, let's say the molecular dynamic simulation for these experiments by Derek that I was describing, right? And now I can feed the forward problem is that I can feed my model with parameters. Given the model, I can feed the model with parameters and I get some data. All right. And this output mapping could be even though the molecular dynamic simulation is stochastic, right? Because it's based on stochastic differential equation. This mapping could be deterministic because I could be measuring some ergodic averages. All right. I could be measuring, for example, the average extension of the molecule throughout the whole simulation. All right. Now, when Derek does the exact same thing, of course, because of errors in his apparatus and so forth, he has some errors in his measurements, which in my modeling description, I just, represent by some Gaussian term over here. So the data that I obtain is the output of the simulation in the forward approach, plus some, let's say, in order to simplify the discussion, Gaussian error. It doesn't have to be Gaussian, but for the moment, let's make it Gaussian to simplify the discussion. All right. And that's the forward kind of problem. Now, the inverse problem is, given the data now and the noise, we would like to invert that relation, all right, efficiently. All right, so we would like, given specific data, to infer the parameters, for example. And you would like to do that even for design. If you want to design one of these devices, for example, so that the molecule kind of extends, it's very expensive to design these devices because, again, these are 100 nanometers apart. You have to do lithography to create these nanowells, etc. So you cannot go about it with trial and error, right? So if you could design them so that you achieve the behavior that you want, you observe some desired data, you infer the parameters that will give you the desired data, then that's something desirable. So if you frame this in the complex of a Bayesian setting, if you think about it, here's Bayes' formula, right? So we have a prior distribution, we have the likelihood, which is the probability of the data given some parameters, and you have the posterior distribution, which is the probability measure of the parameters given the data. Now, the likelihood corresponds to the forward problem. All right, so given the parameters, I feed my model parameters. My model, that's exactly what a stochastic simulator would do for you, can estimate the probability of observing certain data, right, given these parameters. The posterior corresponds to the inverse problem, right? If you want to find the parameters that will give you a specific time series of data, what you would like to do is that you would like to maximize that posterior probability measure, all right? Maximize it over the parameter space, given the specific data that you have observed or that you desire to observe. All right. So there's a lot of discussion here about frequentist approach to estimation and Bayesian approaches to estimation. Basically, frequentists focus more on the likelihood and Bayesians focus more on the posterior distribution. I'm not going to go through this discussion. All right. Ask me about it. Ask me what I mean about this. But the idea is that if you want to maximize the posterior, very early on, you realize that all you have to do is to devise an algorithm that efficiently samples from this posterior distribution. You want a fast sampling algorithm for sampling from this distribution, because if you are able to sample from this distribution efficiently, all right, you can obtain a lot of independent samples, and your samples would come from the regions of maximum probability. So to speak, you will be able to explore the terrain of that posterior distribution. And so how do you sample from a distribution like that? The Markov chain Monte Carlo idea is the following. I'm going to design a Markov chain, and that's not mine, but I'm going to tell you about the improvements of this idea that we did. I'm going to design a Markov chain that has this posterior distribution as an invariant distribution, and I'm going to design the chain in a way so that it's ergodic. So if it is ergodic, it will have a unique invariant distribution, which by design is going to be this posterior, and it will have to converge to that distribution, right? If it is ergodic, if it is designed to be ergodic. All right. So how do you do that? It's very easy to do that. The Metropolis algorithm does this for you. All right. Notice that what you have over here, again, you have access to this likelihood. That's what your stochastic simulator does. And the prior distribution for us is going to be agnostic. It's going to be basically the uniform distribution over a compact domain. We assume that we know nothing. That's a worst case scenario. Nothing about which parameter can give you, the desirable data. All right. So this pi over here is just a uniform distribution over a compact domain. All right. So this is called the evidence over here in the denominator is a high dimensional integral. So that's kind of difficult to compute. So you don't want to be in a position where you want to compute that. That's why we're devising the Markov chain of the Carlo algorithm. All right. So the idea here is that we have something that's proportional to the posterior. It can be evaluated. All right. And the Metropolis algorithm, what it does, this is a 2-minute introduction to the Metropolis algorithm. It says, all right, the x-axis here is a parameter space. So I have a Markov chain that moves around in parameter space. And if at time k, I'm at this position xk, here's what the Metropolis algorithm does. It generates a proposal, another parameter, according to a symmetric distribution, say a Gaussian. All right, suppose that now this new parameter is xk plus 1. And then it compares the ratio fxk plus 1 over fxk, right? F is basically the product of the likelihood with the prior distribution as a function of theta, right? The likelihood as a function of theta, right? These are accessible to you because the likelihood is what you can compute with your stochastic simulation, but they're expensive. You would like to minimize the calls to this function f, all right? So then you compute this ratio over here. If this ratio is greater than 1, then you accept this proposal. If this ratio is less than 1, if it's greater than 1, you accept the proposal. If it is less than 1, p less than 1, then you accept it with probability p, and you reject it with probability 1 minus p. All right, so you go back to xk with probability 1 minus p. Now you can prove that that Markov chain converges to the posterior distribution. All right, provided that this f was defined through the likelihood. All right. The problem with that, so what does this mean? This means that if you converges regardless of the initialization of the Markov chain. The problem with that, so that's a good thing, because if you initialize it and you follow the trajectory of that seed that you use to initialize the Markov chain, then after a number of steps, you will have a good approximation to a sample from the posterior distribution. How many steps? Mixing estimates do exist for the Metropolis algorithm. All right. Now, the problem with that is that the Metropolis algorithm gets stuck. It converges, all right, that's fine, but it gets stuck for long times if your posterior distribution is multimodal. The Metropolis algorithm gets stuck into specific modes. It will eventually converge, right, the whole multimodal distribution. But you see here in 1D simulation, and for 10 to the 5 steps, it's stuck over here. Slow convergence. And you don't like that slow convergence because at any step, you make a call to the molecular dynamics simulator, which is super expensive. So that's really out of the, it doesn't really work for molecular dynamics. All right. So what you would like to do is that you would like to have some alternative approach that takes care of multimodal distributions. And here's the basic idea of the transitional Markov chain Monte Carlo. The idea is that you create a homotopy. Instead of going towards the posterior distribution at once, instead of trying to sample from the posterior distribution at once, you create a continuous deformation from the prior to the posterior. So it's going to be a discretized homotopy. All right, so you're going to have a sequence of intermediate distributions, which are going to be given by this expression. So for qj over here is equal to 0, you have the prior distribution. For qj, the exponent qj over here is equal to 1, you have the posterior distribution. Right, modulo the normalizing factor that you don't want to compute, right? And so the idea is that let's devise an algorithm that samples from these intermediate distributions instead of immediately try to sample from the posterior distribution. Why is this a good idea? This is a good idea because for lower exponents over here, heuristically, that's a good idea. Because for lower exponents in this homotopy, essentially what you are sampling from a distribution that is very close in the total variation distance in the agnostic prior distribution, and the agnostic distribution is uniform, so it samples. It basically kind of spreads your sample, so it discovers multiple modes because it spreads your sample. All right, and as you go through this homotopy, as you increase the exponents, then your samples will start concentrating to the modes. All right, to the peaks. Right, that's a heuristic idea. Now, that's not an algorithm. That's a philosophy behind a number of algorithms that exist out there. So I'm about to show you our implementation of this idea. But the idea is that you create a homotopy and then you try to exploit information on the intermediate distributions. Here's how we do that. Should I tell you how we do that? I have only 10 minutes. All right, so I'm not going to tell you about our implementation. Ask me about our implementation. Our implementation is embarrassingly parallel, all right, which is the only implementation of the team CMC idea. It's actually parallelizable. All right, so I will skip that, but I want to show you how does this work. All right, so here is a toy example, which by design has a bimodal posterior. All right, so we want to design a lipid bilayer, which has a specific curvature. All right, so basically lipid bilayers consist of basically these lipids that have these tails, and the tails come with a harmonic force for the angle of the tails. All right. The idea is that when you pack these lipids in a lipid bilayer, then you have some elastic energy stored because you squeeze these tails, and these tails elastically resist the squeeze. All right, that's the idea. And now you see what happens. If you put different types of lipids on the top and different types of lipids on the bottom, then you will create a curvature, because if you have these guys at the bottom, they would like to expand, and that creates a curvature. That's not a geometry problem, that's a mechanics problem, right? Because the curvature depends on the kind of how strong are these forces, how strong are these harmonic potentials for the angles. All right. So the forward problem is that, all right, I put the lipids, I run a DPD simulation, and I observe the curvature. The inverse problem is that if I want to achieve a specific curvature, what are the angles that I should have over here? That's a toy problem. But it's a toy problem because the posterior is bimodal. Can you see that? Because it has symmetry, right? It doesn't matter. You're going to assume the same curvature, regardless if you have the top angle being this guy and the bottom angle being this guy, or you interchange the top and the bottom angles. You know, a smiley would become a frowny, but basically you can assume the same curvature in different regimes of the parameter space. All right, so again, that's the forward problem. You feed it with the parameters, you get the curvature, but now we feed the curvature to our parallelized TM-CMC method, and we get out the parameters. That's the idea. And very quickly, since I don't have enough time, the idea, let's see here, this is a simulation where basically the top angle was 80 degrees, the bottom angle was 20 degrees. And you see, all right, so we have all these samples here. We explore the posterior distribution. These are samples from the posterior distribution. And you see that we get this symmetry that I just described. If you run this, we have run this with the Metropolis algorithm, it gets stuck to one of these lobes over here. Right? It doesn't move around, it doesn't explode. Eventually it will converge. We have theory that converges, but the convergence is desperately slow. All right, so you can do uncertainty quantification. If the angle is the same, 20-20, then you observe that you should get basically the diagonal, but you see that you get a lot of elements around the diagonal here. And the reason for that is because if you have small angles, all right, if you have large angles, then the membrane is very stiff and it doesn't fluctuate around, because large angles, they're getting squeezed, you have a lot of elastic energy stored, the membrane doesn't move around, doesn't fluctuate. You have small angles, you don't have a lot of elastic energy stored, so the membrane kind of undulates and you get a certain uncertainty in your results, standard deviation around the diagonal, right, which is what you would expect. All right, so this works. And of course, we did, performance analysis. So we compared the method to other methods that exist out there, like Hamiltonian MCMC. Hamiltonian MCMC tries to avoid this problem of the Metropolis algorithm of getting stuck in one basically node by imposing a Hamiltonian that basically sends away the samples from specific nodes randomly. All right, so that you explore. So, we explore the performance of our method in terms of the dimension. These are log-log graphs, all right? So you see here, we explore it in terms of the number of samples. So you have a straight line here in the log-log graph with, a slope of minus 0.5. So in terms of the number of samples, this converges with a rate of 1 over square root of n in terms of the number of samples. Right, and we made comparisons with a number of other methods. It really outperforms, for example, it outperforms nested sampling, if you know what this is. All right, and it outperforms also, obviously, it outperforms methods that are not parallelizable. Hamiltonian Monte Carlo is not parallelizable. All right, so this is parallelizable. All right, we use that on a number of other things. We have used that for fluid flow. One last thing that I'm going to say is that we have used that for Bayesian model selection, if you know what Bayesian model selection is. Because one advantage of our method, I don't have time, I guess, to tell you what this is, but one advantage of our method, ask me about this, but one advantage of our method is that it gives you for free an estimate of the evidence, which is the main quantity that you have. The evidence, the evidence is that quantity over here, is the probability of the data given the model, where you have integrated out all the parameters. That's usually expensive to compute, but our method gives you a pretty efficient estimator for that probability. All right, and there are not many MCMC approaches out there that do that. Nested sampling does that, all right, but we're more efficient than nested sampling. So if you want to do Bayesian model selection, you don't want just to estimate some parameters, but you have a family of models, and now you want to decide which of these models actually has the best fit. This is, for example, it would appear in this kind of scenario where basically you have an aneurysm in an arterial tree and you want to decide where this aneurysm is. So you have a family of models. Every model is parameterized by the position of the aneurysm. And you want to decide which model has the best fit in your velocity data. All right, so our method does that, and other methods do that, but we outperform these methods. All right, so we have, I will give you some of our papers with the comparisons if you want. All right, I think I went very quickly through that, but I think this gives you an idea what we're trying to do here. Basically, we're trying to set up inverse problems for very expensive molecular dynamic simulations. And one approach is the Bayesian approach, but the problem with the Bayesian approach is that inherently it uses sequential algorithms, basically Markov chain Monte Carlo, which is not parallelizable. All right, our approach based on a homotopy, all right, it's highly parallelizable. I didn't explain why, so ask me why. I should say that our implementation of that homotopy idea, right? Again, when you talk about transitional Markov chain Monte Carlo, there are a lot of algorithms out there. TMCMC is not an algorithm, is basically an idea that can be implemented in many different ways. The idea is you would like to exploit a homotopy rather that would spread your sample rather than going directly to the posterior distribution. Our implementation builds on that idea and basically parallelizes that idea, right? So it's pretty efficient. All right, so basically that's what we want to do. We want to design molecular structures on the basis of a Bayesian methodology that would take advantage of experimental data. I went through that super duper quickly. I apologize about that. I think that's a good place to stop. All right, so the next time we have something like 2 or 3 papers together. He's an experimentalist. Clark worked with Derek, and then he set up most of the dissipative particle dynamic simulations. We had very good fit with the experimental data. Karen did, so I spent a sabbatical semester working with Petros, who back then used to be at ETH, now he's at Harvard. Petros is one of these people that I want to bring for a seminar to UC. Petros is by training a fluid dynamicist, a computational person, but when I spent a sabbatical there, we started discussing about the homotopy method and we started doing those implementations. I'm not a high performance computing person. I had to learn this stuff working with him. And so when I went back to Brown, I had Karen started working with this stuff, and now Karen works as a data scientist. I guess that's all. Thanks.
Thank you, Tassos.
Right.
Virtual applauses, yes.
Right, great, thanks.
Any questions from anyone in the audience? Pedro, yes, go ahead.
Thank you, Tassos, for your presentation. I have a couple of questions in regards to just to have a feeling, right? So in terms of the parameters, the first or one of the earliest models that you showed, right, the birth and death process, that one only had 4 parameters, right?
That's right. That was a toy model. Yeah, we wanted to do analysis there. That's why. Yeah.
So how does scale your algorithm when you have like a larger parameter space?
Right. Yeah. So this is this figure over here. So we have a big discussion about that in one of the papers. I can give you the paper, but that's just one of the figures that I had over here. All right. So you see, this is the dimension of the parameter space. Again, this is a log graph, right? So basically, you can get some polynomial. So I don't remember now what the slope of these curves are, right? So suppose that slope over here is 2. It's not 2, but basically that would, because it's log, that would basically increase the dimension squared, depending on the slope of this guy over here, right? And what I have here, this is tested on a setting, not on one of these complicated models. The posterior here is known, so you can basically measure the error, right? So in the blue curve is basically a high dimensional Gaussian, all right, where basically you increase the dimension of the Gaussian, so you increase, the dimension of the mean vector and the covariance matrix, all right. So in dimension 10, the number of parameters would be 10 parameters for the mean vector and 10 by 10 for the covariance matrix. And here you have the mixture is a mixture of Gaussians. So here for this plot, we had 5 Gaussians, not 5 Gaussians, but a mixture of 5 Gaussians, right? A mixture of Gaussians is a distribution by itself. Interestingly enough, for that mixture of Gaussians, actually, it was doing better than the unified distribution was. So that was a little bit discouraging initially because we're tailoring this algorithm to multimodal distributions, like the mixture of Gaussians. All right. But apparently, in applied settings, when you have models like the one that I showed you, not clean things like a mixture of Gaussian, it outperforms. I don't have here the comparison with nested sampling, for example, which is why am I talking about nested sampling? I'm talking because nested sampling is a Bayesian methodology that is parallelizable, like ours. Right, Hamiltonian Monte Carlo is very trendy nowadays, but it's very trendy because you can run it on your laptop. It's very fast if you want a fast, non-parallelizable algorithm. So it's not an algorithm that you can use for this kind of stuff, right? But
in that case, you need to compute Jacobians and gradients when you use the Hamiltonian Monte Carlo.
Right, there are all sorts of things, and I mean, I don't know. I mean, obviously somebody might come and parallelize it at some point, but this hasn't happened yet. All right, so Hamiltonian Monte Carlo is very trendy, but it's like comparing apples and oranges because obviously we're much more efficient because in terms of the implementation because we have parallelized that, right? So
yeah.
So the point now is that if you compare this, you have to compare it with other methods that are parallelizable. So nested sampling is one method that's parallelizable. And there are multiple actually parallel implementations out there. All right. So, and the other similarity with nested sampling is that it gives you estimators for the, I haven't talked about that much, but it gives you estimators for the evidence, which is the main quantity you want when you want to do model selection. I haven't talked about model selection. And we're interested in model selection. We're not interested in just estimating parameters, but we're interested in a family of models, which one is best, right? To have a methodology of deciding the best model, right? So, so we have very extensive computational comparisons with nested sampling, but that is not here, all right? So that's, yeah. It's not in the slide, but it's in the paper. That's what
yes. So in your models for the molecular dynamics, right? At some point, you will have to compute every time the likelihood. And I didn't quite understand if you need to run every time you need to compute the likelihood, you need to rerun your molecular dynamics simulation. How do you do it?
You do.
Right? And
yeah,
that's why will be expensive, right?
That's expensive because you have to do, and that's where I had to educate myself, right? So you have a very expensive molecular dynamics simulation, and you have the Bayesian methodology that we have, implementation that runs on top of it, and both have to be parallelized. So you have to do some very elaborate load balancing in terms of high performance computing, because then you have just some processor sitting idle and doing nothing, and that's not all right. So the important thing for the likelihood, the complexity of computing the likelihood really depends on the statistical model that you adopt. All right, so because at the end of the day, think about it. If you measure with your molecular dynamics simulation some ergodic average over here, so that's a deterministic quantity, and you perturb the output with some Gaussian, right? That's very easy in some sense because you know that then the data are going to be Gaussian with mean zero and variance given covariance structure given by the covariance structure of this guy. So basically, the only thing that you do is that you use your molecular dynamics simulation to compute the mean of the data, right? Sorry, they say the data have a zero mean. No, the data have mean, which is given by this guy over here, the output of your molecular dynamics simulation. And then it has a covariance matrix that is given by that Gaussian random variable here. So that's a very simplistic approach. In the fluid dynamics problem and other problems that we have worked with, we adopt more complicated uncertainty models where basically, I think I have it here, using a misspecified model, where basically we don't only perturb the outcome of the simulation, but we also perturb the parameters, right, with random variables. And so these perturbations propagate in a nonlinear fashion through the evolution of the model, all right? And there, the computation of the likelihood cannot be given analytically. If this is your stochastic model, sorry, if this is your statistical model is basically the output of your simulator plus a Gaussian perturbation, then the statistics of the data are trivial because it's just a Gaussian with mean this guy and variance, the variance structure, the covariance matrix of your Gaussian. All right. Yeah. But so that's, that simplifies the problem a lot, right? So you don't need to have multiple runs of your molecular dynamics simulation to compute statistics and estimate the likelihood like that. Your likelihood is known in some sense. The structure of the likelihood is a Gaussian, and all you have to do is estimate it in the mean. So you need just one run of your molecular dynamics simulation. But if you have more complicated statistical models where the parameters are unknown now, they're perturbed by some Gaussian, then you have to have multiple runs of your molecular dynamics simulation. That's a mess. It requires very elaborate load balancing.
Right.
Right.
So how would this compare, to likelihood-free methods where you somehow you simulate from your model, or you simulate data really? You don't compute the likelihood, and you summarize the data that you simulated using some summary statistics, and then you use that as a measure of your likelihood. Would that improve maybe the efficiency of the methods you use?
Yeah, so that's actually an excellent question, and that's something that I work on now. So think about, and I work on that on a very simplistic setting, because I want to do analysis on that. There is a little bit of analysis already. So think of a single stochastic differential equation, the Langevin equation or whatever, right? And you form, you set it up in a weak setting and so forth, right? Appropriately. And now the idea is that you would like to apply a Bayesian setting to that. Now, the problem with that is the uncertainty now is driven by the stochastic differential equation if you don't take any ergodic coverage or whatever, right? So it's definitely not Gaussian. It doesn't have to be Gaussian. It's going to be some functional of a Gaussian, because again, the stochastic differential equation will be driven by white noise, right? So some Gaussian process, but your data would be some function of that process. All right, and so you get something highly non-Gaussian. The likelihood there cannot be, you don't have specific knowledge for the likelihood that you have here. And in a likelihood-free method, what you do is that you say, all right, so I'm going to track the numerical scheme that I'm using for that stochastic ODE now. And by tracking that, I'm going to find some estimators of the likelihood by the evolution of the numerical scheme. All right, and so that computationally, if you do it, it kind of simplifies things. It seems to be working very nicely, but nobody has analyzed. There are a couple of papers that analyze that. There are a couple of papers out there that analyze that with ergodic theory for a very simple, a single stochastic differential equation, not a system, right? But there's a lot of work to be done here. And so 2 comments on that. I haven't applied these in this methodology. I don't know how well it would play with the fact that you have a very high dimensional system. It could play well, but I haven't done it. I haven't implemented. But I also believe there are very good opportunities there for some very feasible analysis. And the steps, in some sense, have already been undertaken, right? So you use basically ergodic theory to provide some asymptotic results there to show consistency of the estimator, for example, by means of ergodic theory.
Yes.
So that's actually really that's something that I'm very interested in. We haven't done this here for this molecular dynamics simulation.
Right. I mean, because it's very difficult to apply full Bayesian on any type of situation because of all the difficulties you
don't have the likelihood. Right? Exactly.
Yeah,
you don't have the likelihood,
and really when you can, that's perfect. But oftentimes you simply can't, because it's just simply too expensive computationally, and then you have to resort to some strategy, right, to simplify or maybe approximate what you really want to compute. The last question: have you considered doing or applying for larger models or more complex models variational approaches rather than just sampling? Define and optimize and find the parameters of your posterior, and by optimization.
Right. So variational, So variational approaches are not something that I have a great experience with. I'm very familiar. I have taught variational approaches, but it's not something that it's feasible in the scenario that I just described, where basically you have an ergodic, your measurement here is an ergodic average, and so you have a good knowledge again of the likelihood. All right. So in a situation now where you perturb the parameters in here, you have a nonlinear perturbation where you perturb the parameters in here, etc. I'm not, but that could be my inability. I'm not saying that it cannot be done. It's just that I'm not an expert in this approach. I'm not sure how to set up a variational approach when I perturb the parameters in the model, rather than, it can certainly be done for a statistical setting like that, where basically you have a deterministic average and you just perturb the deterministic average with a Gaussian, and then you can have a variational kind of approach.
I was just wondering, I mean, because it's apparently for large scale problems, it may scale better. I'm not an expert either, but I was wondering whether you have some experience anyways, I mean, we can talk about it later. Thank
Right.
you.
Right.
That was very interesting, your talk.
Yeah, thanks. Yeah.